Thank you, Irfan, for the kind introduction. Thank you all for joining us today. On behalf of myself and my co-session lead, Dr. Ray Kantav, a Senior Regulatory Project Manager in the Office of Quality Assurance within OPQ, welcome to the final session of the Advancing Generic Drug Development Workshop. The session is titled Nitrosamines, Known Issues and Practical Advice. We hope that you'll find this session brings the workshop to a close on a high note. In the session, we'll hear 3 presentations discussing the challenges generic drug developers are facing with nitrosamine impurities and providing practical recommendations to overcome these challenges from bioequivalence, quality, and pharmacology, toxicology perspectives. From a bioequivalence perspective, Dr. Gong Zhao will present nitrosamine-impacted drug products containing biopharmaceutics classification system, or BCS Class IV drug substances. Dr. Zhao is a research fellow in the Division of Therapeutic Performance II within the Office of Research Standards in OGD. From a quality perspective, Dr. Matthew Vera will present Addressing Potential Pitfalls in Nitrosamine Risk Assessment and Control. Dr. Vera is a supervisory chemist in the Division of Product Quality Assessment II within the Office of Product Quality Assessment I in OPQ. Finally, Dr. Xin Fu will present Pharmacology, Toxicology, Considerations for the Safety Evaluation of Nitrosamine Impurities. Dr. Fu is a pharmacologist in the Division of Pharmacology, Toxicology Review within the Office of Safety and Clinical Evaluation in OGD. And just let everyone know, following the presentations, we'll immediately transition directly into a Q&A panel, providing an opportunity to engage with subject matter experts on these topics. And in that, our presenters will be joined by 2 additional panelists, Dr. Joanne Chia, the Director of the Division of Product Quality Assessment 10 within the Office of Product Quality Assessment 2 in OPQ, and Dr. Rong Wang, the Deputy Director of the Division of Bioequivalence 1 in the Office of Bioequivalence in OGD. With that, I'll ask Dr. Zhao, to go ahead and kick us off.
Thank you, Chris, for the nice introduction to all the presenting authors. And it is my great honor to present the nitrosamines in this session 4, nitrosamines known issue and practical advice. So the title of my presentation is Nitrosamine-Impacted Drug Products Containing BCS IV Drug Substance. My name is Gong Zhao. I'm from Division of Therapeutic Performance II, Office of Research and Standards, Office of Generic Drugs. So first of all, disclaimer, this presentation reflects the view of the author and should not be construed to represent FDA's view or policy. So the outline of my presentation. My presentation will cover the current nitrosamine challenges across immediate release oral products with various BCS classifications, and FDA's alternative bioequivalence framework and the BCS Class IV limitations. I'll also cover the proposed risk-based subcategorization for BCS Class IV drugs and a case study to validate the regulatory implications. So these slides illustrate the distribution of the nitrosamine-impacted immediate release oral drug products. As shown in the left pie chart, we have identified 160 nitrosamine-impacted drug products among these IR products we collected, which represents about 13.88% of the IR products we collected. And the red pie chart shows the distribution of the BCS classifications of the nitrosamine-impacted oral IR drug products. And we have about 46 of the BCS1, which count about 28%, and the 24 in BCS Class 2, and 58 in BCS Class 3, and about 32 in BCS Class 4. Let's examine the characteristics of these nitrosamine-impacted drugs. The chart on the left shows the food effects of the nitrosamine-impacted drug products across different BCS classification. And the right chart shows the fraction absorbed of the nitrosamine-impacted ROI drug products. And as we can see in the BCS class 1 and class 2, the majority is the with the high fraction absorbed, over 85% of the fraction absorbed. And the BCS class 3 and the BCS class 4 has the moderate or low fraction absorbed, which is between 50 to 85% or lower than 50%. So now let's review the current FDA guidance on alternative B approaches for nitrosamine-impacted drug products. So the guidance focuses on the alternative B approach for products with the added antioxidants and the pH modifiers. For reformulated products to include excipients such as antioxidants or pH modifier aimed to reducing nitrosamine impurity, the FDA guidance allows for the use of alternative B approach to demonstrate BE without the need for traditional in vivo and fasting or fat condition B studies. The guidance also highlights the importance of BCS classification, which is central in determining alternative B approach. For BCS class 1, class 2, and class 3 nitrosamine-impacted RIR products, the comparative dissolution under multi-pH dissolution condition may be used as an alternative B approach. If the reformulated IR products use pH modifier or the specific antioxidants like listed in the guidance, ascorbic acid, alpha-tocopherol, cysteine hydrochloride, or the propagylate within the acceptable limits, typically lower than 10 milligram per dose. However, for BCS class 4 drugs, the guidance recommends more advanced approaches, such as studies with the validated in vitro in vivo correlation, physiologically based pharmacokinetic modeling, or in vivo B studies. So the scientific justification and the risk-based approach when selecting alternative B approaches. The guidance recommends a thorough risk assessment considering the factors such as drug properties, nitrosamine formation pathway, and the extent of the formulation change. Now let's examine why BCS IV drug products present such unique challenge. The first of all is the BCS class 4 drug products has a characteristic of absorption complexity. The BCS class 4 drug substances are characterized by both low solubility and permeability, making bioavailability or bioequivalence unpredictable and challenging. The comparative dissolution test may not reliably predict complex absorption behavior, particularly compared to their effectiveness with more soluble and permeable drug. The BCS class 4 drug also has their formulation sensitivity. Due to the intrinsic challenges to BCS class 4 drugs, even small formulation changes may necessitate rigorous B evaluation methods to ensure that the therapeutic performance is maintained. However, I believe there is a pathway forward with a robust risk-based approach, including rigorous dissolution testing and PBPK modeling methods, when necessary, can provide a pathway to waive the in vivo B studies. This approach is particularly applicable for BCS IV drugs when changes involve adding the antioxidant or pH modifiers, which has been listed in the guidance for other BCS class drugs, and with limited impact on the overall absorption. So this brings us to our proposed solution. This table presents a classification of BCS IV subcategories along with their associated risk factors and critical excipients, it breaks down BCS4 drugs into different subcategories based on their dominant limitations, whether it is solubility limited or permeability limited. And it identifies the corresponding challenges relevant to each category. So if it's solubility limited, the BCS4 drug products may behave like BCS2 drug products. And the risk factors including the pH-dependent solubility or precipitation. And usually the food effects will be positive food effects with higher fraction absorbed. And the critical excipients in these drug products may contain pH modifiers or polymers or solubilizers. For permeability limited BCS4 drug products, it may have similar behavior as the BCS class 3 drug products, which has a low passive permeability or efflux transporter or has a short absorption window. And usually the food effects of these BCS4 drug products usually has no food effects or negative food effects with a lower fraction absorbed. And the critical excipients usually include surfactants or permeability enhancers. So in between is those BCS4 drug products which has the dual limitation. They're both limited by the solubility and the permeability. And usually for these drug products, the food effects is unpredictable. So building on this subcategorization system, we propose a systemic 4-tier risk evaluation framework that provides the graduated evaluation based on the risk level and the drug substance characteristics. So for solubility limited drug products, usually the key indicator is positive food effects. And by conducting the solubility pH testing, we can identify the solubility limited BCS4 drug products. And for permeability limited, the usually key indicator is no or negative food effects. And we can evaluate the estimated fraction absorbed and comparing to the known model drugs in the BCS class 3 as listed in the guidance, and we may be able to extend those findings to BCS class 4 drugs. And the dissolution testing is also the key. By conducting the comparative dissolution testing in QC or multi-pH media, we can determine whether it is solubility limited or permeability limited. And the key indicator usually is dissolution not similar or the more advanced absorption prediction PBPK modeling suggests high risk. Then if it will be studies will be recommended. So the scientific foundation of our approach comes from this FDA extensive research. So FDA conducted the research of these model drugs, including the cimetidine, acyclovir, atenolol, ranitidine. And they got the estimated fraction absorbed of these model drugs and also the food effects of these model drugs. So the research on the BCS3 model drugs show that with the addition of the antioxidant do not alter the permeability. So for BCS4 drugs with comparable or better permeability or absorption than those BCS3 model drugs, antioxidants are expected to have minimal or no effect on the permeability. So for BCS4 drugs where permeability is not the primary limiting factors, antioxidants is not expected to affect drug permeability and absorption. In more complex BCS4 formulation, it's essential to confirm that the antioxidant amount threshold, such as if it's over 10 milligrams, do not interfere with other excipients, such as solubilizers or surfactants, or introduce permeability alteration. So next, let's take a look at these hypothetical case studies. So in this case study, the drug product is IR tablets with multiple strengths, and it's a kind of antibiotic. The nitrosamine impurities of this drug product was detected in all the tested batches of the marketed products. And the nitrosamine impurities level exceeded the acceptable intake limits of 1,500 nanogram per day. By analyzing this nitrosamine impurity level in this drug product, the root cause is coming from nitrate impurities in the excipients, and also the manufacturing process conditions, and also the nitrosamine will form during the storage condition during the shelf life. Let's take a look at the nitrosamine risk mitigation strategies for this case. So this drug product utilized the reformulation by adding the antioxidant, which they used the level less than 10 milligram per dose as the nitrate scavengers through the pre-approval supplement. This table shows the formulation composition before the reformulation and after the reformulation. So the API, disintegrant, binder, extragranular stays the same, and they added the nitrate scavenger in the reformulated product and adjusted the diluent to keep the same weight of the tablet. And the risk assessment shows this drug product is a low risk per the ICHM 13A guidance, which it does not have a special formulation or a special manufacturing technique. Let's take a closer look at the characteristics of this drug product. So the maximum single dose of this drug product is pretty high, 780 milligram, and it is a BCS class 4 drug product. It has a pH-dependent solubility, which the solubility is very high until pH 6, and at the pH 6.8, the drug substance precipitate, and per the ICHM 9 guidance, it cannot be claimed as a high solubility drug substance. The drug product has a moderate to high permeability with about 68% to 72% absolute bioavailability. And the food effect is a little bit negative to minimal no effect. So the AUC under fat condition dropped about 25 to 20% comparing to fast condition, and the Cmax dropped about 25 to 30%. So this slide shows the dissolution profile before the reformulation and after the reformulation. So before the reformulation, the dissolution using the QC media showed very rapid dissolution. As we can see, 98% were dissolved within 10 minutes. And after reformulation, it shows very similar profile, and 93% dissolved after within 10 minutes. So this drug product also meets the very rapid dissolution criteria, which over 85% dissolved within 15 minutes. So the PK profile is very persuasive as well. So before the reformulation, the TR ratio under fasting condition and under fat condition is 95 and 105% of the AUC. And the Cmax shows similar trend. Under fasting condition, the TR ratio is about 90%. And under fat condition, the TR ratio is about 102%. So the PK profile after the reformulation, the TR ratio seems tighter comparing to before the formulation. So it shows about 99% under fasting condition and 100% under fat condition. And it's similar for the Cmax as well. So with the in vivo PKB studies, the antioxidant addition did not impact BE, confirming the predictable antioxidant effects similar to established BCS class 1, class 2, or class 3 approaches as listed in the guidance. So this case study demonstrated several key principles that validated our proposed framework. First, the antioxidant addition of the less than 10 milligram per dose, maintained the bioequivalence, supporting the theoretical basis of predictable antioxidant impact that we derived from the BCS model drug research. From a framework relevance perspective, this case shows the high solubility up to pH 6 and moderate to high permeability with no significant food effects, supports the full-tier risk-based framework, leveraging the established BCS class 1, class 2, and class 3 approaches. So this case study also suggests the feasibility of BCS IV drugs with similar absorption characteristics and reformulation strategies. However, we recognize that future research needs to include additional BCS IV cases to refine and validate the proposed framework and continued evidence generation for alternative B approaches across different BCS class 4 subcategories. Let me conclude with the key considerations and the future directions. For nitrosamine-impacted drug products, it is critical to ensure that formulation change made to control nitrosamines do not affect solubility, permeability, or in vivo bioavailability and bioequivalence. Food effects are critical in the subcategory classifications. Positive food effects with higher fraction absorbed often indicates solubility limited, similar to BCS class 2 behavior, while the low fraction absorbed with negative or minimal food effects suggests permeability limitations similar to the BCS class 3 behavior. The predictability of the antioxidants effects allow for tailored BE assessment for BCS 4 drugs. Research on the BCS 3 drugs show that antioxidant has minimal or no impact on the permeability, findings that can be extended to the BCS 4 drugs with comparable or better permeability. They established the alternative B approach for BCS class 2 and BCS class 3 can guide BCS class 4 B assessments. For future research, particularly for the dual-limited BCS class 4 drugs, we should focus on the complex formulation in the presence of antioxidants, pH modifiers, and solubilizer permeability enhancers. This includes assessing the existing PK data, determining how these excipients affect the absorption dissolution across pH ranges using the QC dissolution method, and investigating how these excipients affect the solubility in complex formulations. We are cross-validating the findings with in vivo data to address the formulation-related risks. Last but not least, I would like to thank my office director, Rob Lamberger, who is the pioneer of this risk assessment framework. And I would also like to thank Lei for her support, and my team lead Chi from DTP2, and also my DTP2 colleague Halim E, and DTP2 leadership Dongmei and MJ. Next, please welcome Dr. Matthew Weira for his presentation. Thank you.
Thank you, Dr. Zhao, and good afternoon, everyone. Thank you for joining us, and thanks to the organizers for inviting me. Sound is okay? Okay. So I'd like to start with the standard disclaimer that the views here are mine and should not be construed to represent FDA's views or policies. So to begin the talk, I'd like to give a little bit of an introduction to this issue from the pharmaceutical quality perspective. Nitrosamines can be present as impurities in both drug substances and drug products. And the reason that we're concerned about them is because they are potentially genotoxic agents, and some have been classified as probable or possible human carcinogens. So nitrosamines form chemically by reaction of two precursors, a secondary amine precursor and some sort of nitrosating agent precursor. And wherever and whenever those two precursors encounter each other, under the right conditions, it's possible that they could react to form a nitrosamine. So FDA's current thinking on this issue is outlined in the 2024 guidance, Control of Nitrosamine Impurities in Human Drugs. And also there's a lot of useful information on the CEDAR nitrosamine impurity acceptable intake limit website, which also includes links to the FDA's recommended acceptable intake limit, or RAIL, guidance as well. So the control of nitrosamine guidance lays forth this sort of three-step framework for managing this issue. The first step is to perform a risk assessment. The risk assessment should cover the risk that nitrosamines could end up in a drug substance or drug product. That doesn't have to be submitted to the agency, but it should be available if requested. The second step then is that if there's any nitrosamine risk identified, you should perform confirmatory testing to establish whether or not nitrosamines are actually present and at what levels. And then finally, if there are changes needed to ensure that those nitrosamines are present at or below acceptable levels, those changes should be implemented and reported as well. And as I said, this is described in the control of nitrosamine guidance. So in terms of nitrosamine risk, I think it's important to point out that there are a couple different kinds of risk to think about. The first one I would call contamination risk. So contamination risk is the situation where those two precursors have already found each other at some early stage in perhaps a raw material or a drug product component and already formed a nitrosamine. So this risk is typically examined and assessed through raw material testing and looking for nitrosamine content in things that are coming into the manufacturing process. Formation risk is something a little different, and I think this is more complicated. The reason it's complicated is because those two chemical precursors may be encountering each other for the first time in the manufacturing process of the drug substance or drug product or during shelf life. And so the precursors may be coming from different places. So testing components for nitrosamines doesn't really shed any light on this. You really need awareness of what possible precursors you have and where they are in your manufacturing process and formulation. So the prototypical example of a formation risk would be the NDSRIs, the nitrosamine drug substance related impurities, where the amine precursor is the API and the nitrosating agent precursor is usually some residual impurity in some other part of the drug product, often an excipient. So with that as introduction, I'd just like to read through quickly the common pitfalls that I want to spend the rest of the talk addressing in a little more detail. The first is not considering nitrosamine risk holistically during the risk assessment stage. Secondly, there can be issues with nitrosamine reference standards, acceptable intake limits that are not endorsed by FDA, misunderstanding interim limits and what that means and how they're used. The issue of how to handle control of multiple nitrosamines in the same product. And then also options that are available when a product or a nitrosamine in a product exceeds the acceptable intake limit. And then finally, just the fact that the understanding of this issue has changed so quickly that it can be a challenge to keep on top of. So the first pitfall, as I said, is not considering risk holistically. So every risk assessment should start with a risk question. This is a concept from ICH Q9 that defines the risk question as the problem statement or the concern that the risk assessment is trying to address. So one of the ways that this can be a problem is if the risk assessment is only considering contamination risk and focusing on testing incoming materials, it may miss formation risk, where there is one precursor in one component and another precursor in a separate component that encounter each other for the first time once you start manufacturing. So in addition, even if one is considering formation risk and NDSRI formation, there are really two different types of NDSRI formation. There's what I would call the classical NDSRI and then the fragment NDSRI. And we'll talk a little bit in a moment about each of those. So the Cedar Nitrosamine AI website shows a list of APIs that have a hypothetical, not necessarily confirmed, but hypothetical risk of nitrosamine formation. So the classical NDSRI scenario is the situation where the pure API itself is a substrate for nitrosation. And this is best exemplified, I think, by bumetanide, which is a very reactive substrate for nitrosation and can quite readily form nitrosobumetanide in formulated products. So some of my OPQ colleagues have published a study of this exact example showing how nitrosobumetanide can form and also some ways to inhibit that formation. So this is a good example of the classical scenario. The fragment NDSRI scenario is chemically the same, but different in one important way. The difference is that in the fragment scenario, the API itself is not a substrate for nitrosation, but an impurity within the API is potentially a substrate for nitrosation. And there's a study of this exact example with lidocaine published very recently, also in JPS, that shows how this can be investigated and understood. So again, in not looking at risk holistically, you might look at the structure of lidocaine in a risk assessment and say, that's not a secondary amine. It's not going to be a substrate for nitrosation. There's no NDSRI problem. But there is the issue of this impurity that could lead to a separate NDSRI. Pitfall number 2 involves issues with nitrosamine reference standards. So if you've gone through this process and done your risk assessment and decided that you need to perform confirmatory testing, you're probably going to want an authentic reference standard of the target nitrosamine so that you know what you're looking for and you'll know whether or not you're seeing it. And if you are seeing it, you're going to want to be able to quantitate it. So a standard is important to have. The problem is that certain reference standards may not be available commercially. And even if you find a commercial supplier, there are cases where suppliers are providing reference standards that in fact aren't the right structure. We've seen that happen a couple of times. So wherever you're getting a reference standard, it's important to characterize it thoroughly and make sure that it is what you think it is, because you don't have time, obviously, to go chasing ghosts, right, for some nitrosamine that in fact doesn't exist. And I think this difficulty stems from the fact that certain secondary amines are just very difficult or impossible to nitrosate. This includes some of the hypothetical NDSRIs that are listed on the CEDAR website. So it's important to realize that if you're trying to source or make a reference standard for a nitrosamine and you're finding that you can't do it, that's an important finding for your risk assessment, right? Because if you can't intentionally nitrosate an API chemically in a flask under synthetic conditions, then it's probably a pretty low risk proposition that nitrosamine would form on its own in a drug product during shelf life, okay? So the guidance specifically says that in situations like this, the inability to make a reference standard or intentionally nitrosate can justify omitting confirmatory testing. So if you find that situation, if you find yourself in that situation, how would you evaluate the susceptibility to nitrosation? That's a very chemically intensive question, and there are several important factors to consider. The reaction solvents that you're using, the pH of the reaction mixture, the basicity of the amine that you're trying to nitrosate, the nitrosating agent that you're using, the possibility that some reaction other than nitrosation could occur, or the fact that the nitrosamine may form transiently, but then quickly react further or rearrange to something else that's also not a nitrosamine. And these last 2 points, again, come back to that issue of characterizing your reference standards so that if you think you have a nitrosamine, you know for sure that it is a nitrosamine that you're testing for. So all of that's to say that because of these multiple issues, the lack of nitrosation in a single experiment is generally not sufficient to demonstrate that it can't be done. So I think a better way to approach this is to think more in terms of a study of a panel of different types of nitrosation conditions to cover these relevant factors. So fortunately, there's some good published literature that talks about all of this. The first paper by Chaudhary is an organic synthetic paper that talks about mild conditions that are generally useful for forming nitrosamines. The Sharma paper uses something called a modified NAP test, which is an optimized procedure for screening drugs for nitrosation susceptibility. And this looks particularly at the issues of reaction solvents. The third paper by Ashworth provides a set of standard kind of complementary force degradation conditions that can be used to understand whether a nitrosamine could form or not. And that paper also discusses the issues of chemical reactivity that I mentioned, the fact that a nitrosation reaction might lead to something other than a nitrosamine. So the third pitfall concerns the situation of AI limits that may not have been endorsed by FDA. So the Cedar nitrosamine AI website has a lot of acceptable intake limits listed, but there may be some that are not listed. So if you find that you're intending to use an AI limit for a nitrosamine that's not listed, that's something that the agency would need to evaluate. Maybe it's a nitrosamine that you've identified for the first time. Or it may be a situation where you're pointing to a higher AI limit that may have been accepted in another regulatory region. Again, that's something that we would have to evaluate here at the agency. And so particularly with approved products, if you're proposing a new AI limit or a higher AI limit than what the agency has endorsed, that really needs to be submitted as a prior approval supplement rather than a changes being affected supplement to allow review time to evaluate that limit. So once we've settled on an AI limit, whatever it is, this graphic at the bottom shows how the AI limits work for control, right? So if you find that a product consistently measures at less than 10% of the AI, no specification is needed. Above 10%, a specification should be adopted. And of course, higher than the AI limit would need either some kind of justification or some sort of mitigation, some way of forming less nitrosamine in the product. So before I get into the mitigation, I want to say a word about misunderstanding interim limits, which is pitfall number 4. So this is just a snapshot of some text from the CEDAR AI limit website that talks about interim limits and how they're used to prevent drug shortages for products that are already in distribution. So if you find that you're worried about being able to supply a product that you're already commercializing, please reach out to the CEDAR drug shortage staff and seek some advice from them on what to do. But the bottom line is that we cannot ever approve an interim AI limit in a specification because it's not considered acceptable, right? So this is a measure that's used on a temporary basis to prevent drug shortages. And as I said, the drug shortage staff at that mailbox can provide further advice. So pitfall number 5 is the situation of needing to control multiple nitrosamines in the same product. So the FDA recommended AI limits correspond to individual nitrosamine impurities and are really only applicable if a drug product contains a single nitrosamine. When more than one nitrosamine is present, we really need a limit for total nitrosamine content. And the control of nitrosamine guidance talks about this in a couple of different places. And I've shown in this snapshot a kind of formidable looking mathematical statement that's in the guidance. And I'd like to try to explain the thinking behind this. So if you take a step back and think about what an acceptable intake limit means, this is a concept that's defined in ICH M7 as a level that approximates an increased cancer risk of one additional case in 100,000 subjects under a conservative set of assumptions involving daily exposure over a 70-year lifetime, okay? So control of total nitrosamines postulates the following. That acceptable level of risk of 1 in 100,000 additional cases could be distributed among more than one nitrosamine. That's the bottom line concept here. So the risk from multiple nitrosamines is additive. And so the aggregate risk from multiple nitrosamines should still be no more than 1 in 100,000 additional cases. So I think a good way to think of this is in terms of percentages of acceptable risk. We don't want any more than 100% of that acceptable level of risk distributed among all of the nitrosamines that we're considering. So let me try to illustrate. Consider a hypothetical drug product that could contain both an NDSRI and some small molecule nitrosamine, like NDEA. And let's assume that there's 100 milligram per day maximum daily dose to keep the math easy. So if those two nitrosamines have the concentration limits shown, and the NDSRI measures at 12 parts per million, okay? That's 80% of its AI. So 12 is 80% of 15. And likewise for the NDEA, if the result is 0.212 parts per million, that's also 80% of its acceptable intake. So both of these nitrosamines are under their acceptable intake limits. But when you consider them additively, right, when you consider the risk additively, we're at 160% of the acceptable risk. And so this is not a result that we would consider acceptable in this example. So there's another more detailed example in the control of nitrosamine guidance in Appendix C, which I'll just briefly point to here. So in this case, there are 3 nitrosamines, and we're looking at a hypothetical stability study. So each AI limit has a concentration limit and a result. And the result is recalculated in terms of percent of the AI limit. And so at the 0-month time point, you can see that the percent of AI limits sum up to 46.5%, which would be considered a passing result. At the 3-month time point, everything has increased a little bit. And when you sum the percent of the individual AI limits, you reach 104.9, which we would consider a failing result. So this is discussed in more detail in the control of nitrosamine guidance. So pitfall number 6 is the situation where a nitrosamine is exceeding its AI limit, and what would be the options in that case? So one option is to justify a higher limit using your own safety data or whatever justification, or alternatively to make changes to the product or manufacturing process or raw material to inhibit or reduce the level of nitrosamine that forms in the product. And these 2 papers are published by some of my OPQ colleagues that looked at this exact sort of effort, both for bumetanide and for metformin, which can form NDMA in certain circumstances. And so in both cases, the studies looked at different levels of different antioxidants, as Dr. Zhao was mentioning in his talk about reformulations, and also a pH modifier, which in some cases can be quite effective as well. The final pitfall that I would just mention is the fact that this landscape is changing so quickly, right? Over the last 8 years, a lot has happened in the nitrosamine area. And so there are some good published articles that I think are useful to understand what's been happening. The first, by Moser, is a study of nitrosamine formation in pharmaceutical solid drug products. And I think that's very important because most of the classical nitrosamine chemistry was studied under solution conditions, sometimes decades ago. And so in the pharmaceutical world, we're looking at behavior in solids, which can be quite different. The second paper by Dirat and his colleagues looks at how several large pharma companies handle the drug substance and drug product risk assessment exercise for nitrosamines, the questions that they ask, the order that they ask them. It's a very good explanation, I think, of how you can approach this sort of a risk assessment question. And then the third paper is a review article by several regulators from around the world a few years ago that summarizes what was known, at least in 2023, about root causes and risk factors for formation of nitrosamines in drugs. So in addition to those papers, I think the CEDAR nitrosamine AI website has some other useful resources. This is a snapshot of the landing page that has this blue button that you can use to subscribe for email updates so that you get a notification whenever something here is changed. There's also some regulatory updates. This is one from June of this year that talks about the revision of the deadline for addressing nitrosamines in approved products. So essentially, we had anticipated having this addressed in August of 2025, but we've realized that isn't going to be achievable for every approved product. And so the current recommendation is that companies submit a progress update by August. The progress update should include the following information. I'll let you read through this in bullet point form in the interest of time. And this information should be submitted in CTD section 1.13.14 of an annual report so that we know where to look for it. And then finally, the AI limit webpage also has some updates on other emerging and scientific and technical issues. There's one for leachables from infusion bags. There was one earlier, several months ago, on ritonavir. So with that, I will stop and thank you for your attention and offer apologies to Dr. Fu for being a little long-winded and going over time. Dr. Fu?
Thank you, Dr. Vera. Hi, good afternoon, everyone. I hope you enjoyed the last two great presentations as much as I do. I found they are very informative. And as Dr. Morgan introduced, I am a Pharm Tox reviewer in AGDD. So my talk will be Pharm Tox considerations for safety evaluation of nitrosamine impurities. Can you hear me okay? So my presentation does not present FDA's will policy, only myself. So my talk will provide a quick overview of nitrosamine and the safety assessment. I will describe how Pharm Tox conduct nitrosamine safety assessment under the FDA guidance and through collaborations. And then after that, I will talk about some common pitfalls from nitrosamine safety assessment from our Pharm Tox reviews. And at the end, I will present 2 case studies. So nitrosamines are potent rodent carcinogens and proper human carcinogens. This finding is mostly based on studies of small molecule nitrosamines, such as NDEA, NDMA, and several others. And currently, actually, the major challenge comes from complex nitrosamines, or also called NDSRIs. Structurally, they are unique to the drug substance and often are larger nitrosamines compared to small molecule nitrosamines. They may contain both activating and deactivating features, which may affect their metabolic activation and potentially mutagenic and carcinogenic potency. And also, we don't have carcinogenicity data for those complex nitrosamines. So nitrosamines as whole belong to cohort of concern under ICH-M 7. Their carcinogenic potency vary greatly in an order of several magnitude. As Dr. Vera's presentation indicate, we use compound-specific acceptable intake to measure their potency and also set a safety limit. So AI limit is essentially an allowable exposure for 1 per 100,000 actual cancer risks over lifetime exposure. The AI limit for nitrosamines are generally less than the threshold of toxicology concern TTC of 1.5 microgram per day for lifetime exposure set by ICH-M 7. So currently, FDA recommend 3 methods to determine AI limit. The first is using carcinogenic potency categorization approach, CPCA, which is basically using structure activity relationship concept to determine the activating and deactivating factors and give them the numerical scores, which is corresponding to 5 categories and 4 fixed AI limit. From 26.5 nanogram per day up to 1,500 nanogram per day. And the second approach is using compound-specific data. This can be carcinogenic data or the mutagens data, which is used to justify the CPCA-based AI limit. The third approach is read-across approach using the surrogate. So the surrogate is basically the compound with sufficient structural similarity with subject nitrosamines. Then the surrogate's AI limit can be used to the subject nitrosamines. So as Dr. Vera's presentation indicate, FDA published several guidance to support nitrosamine risk assessment, including the quality guidance for control of nitrosamine impurities in human drugs, and also safety guidance for recommended AI limit for NDSRIs. We refer them often internally as the real guidance. And also there's the FDA webpage for nitrosamines, which published recommended AI limit for NDSRIs and nitrosamines, as well as many important informations. So for Pharm Tox, we conduct our nitrosamine safety assessment and review following the real guidance as well as information described in FDA webpage. The real guidance described the framework for setting AI limit using the CPCA approach as a starting point when there's no compound-specific data. Only the structure of the nitrosamine is needed. The real guidance also describes the test for nitrosamines and also encourage mitigation approach. Also, real guidance describes options to support alternative AI limit and clarifies expectation for what data are needed to support an AI limit up to 1,500 nanogram per day. In addition to negative enhanced AMIS assay, confirmative in vitro mammalian cell mutation assay and in vitro metabolism studies are recommended. To support AI limit above 1,500 nanogram per day, additional negative in vivo mutagenicity assay are also recommended. And also I want to point out, a negative in vivo mutagenicity assay not necessarily support an AI limit equal to ICH Q3A, Q3B, QT level automatically. The real guidance also describe the surrogate-based AI limit, and the surrogate should be structure relevant with robust carcinogenicity data. And also Dr. Vera described the information in the FDA webpage, and this webpage is established to improve the communication with applicants so that they can get the updated information from recommendation from FDA. It includes sections on emergent issues, the timeline for implementation, and options for approved product when there are drug supply concerns. The website also encourages the applicants to directly contact the FDA when they have a drug shortage concerns. So while we conduct nitrosamine safety assessment through guidance, we also through collaborations. Within FDA, multidisciplinary experts, they form task force to support nitrosamine risk assessment, advance nitrosamine science, and develop nitrosamine guidance. Within this task force, the Pharm tox expert in OGD and OND, as well as in computational toxicology group, they form a nitrosamine safety team to coordinate the nitrosamine safety assessment. The expert in this team participate in Nitrosamine International Technical Working Group, as well as ICHM7 nitrosamine addendum development. They collaborate with FDA's Research Center, NCTR, on research needs for nitrosamine risk assessment. They also support FDA nitrosamine research and collaboration through work with HACI. So the safety assessment of nitrosamines in both new and generic drugs follows FDA guidance on nitrosamines. In OGD, Pharm tox also collaborates with OND to align on assessment approaches. The Pharm tox assessors also work closely with other disciplines to support risk assessment and maintain access to medically necessary drugs. So I described how FDA conduct the nitrosamine safety assessment through guidance and collaboration. Now I would like to discuss some common pitfalls identified in nitrosamine safety assessment submitted for review. The first one is inadequate enhanced AMIS assay. Enhanced AMIS assay is probably currently the most important and commonly submitted assay to support nitrosamine safety assessment. It evaluates mutagenicity potential using in vitro bacteria models. And real guidance recommend to use pre-incubation method and also along with use hamster S9 at 30%. With the publication of real guidance, as well as several publications on this topic, we do see improvement for the quality of submitted enhanced AMIS assay. But we still see some common deficiencies. For example, low text dose tested in the assays. Despite that, without even testing the cytotoxicity at a higher dose level. And for their standard for AMIS assay recommended, the top dose should be 5,000 microgram per plate. But we do see some assays just testing the level they want to propose to control in their product, which is not sufficient because AMIS assay is an assay for hazard identification. You do need to test adequate dose to really truly test the mutagenicity potential. We also see improper selection of solvent. Either the concentration is too high would somehow the solvent interfere with metabolic activation and also sometimes can cause severe cytotoxicity. And also see lack of appropriate nitrosamine policy controls and insufficient demonstration of metabolic activity for S9 use in the assay. Another common deficiency is justifying an alternative AI limit of 1,500 nanogram per day solely based on negative enhanced MSA. As I just mentioned, FDA also recommend additional studies, including in vitro mammalian mutagenicity assay and in vitro metabolite studies, with focus on alpha hydroxylation and the formation of a DNA reactive metabolite, in addition to enhanced MSA assay. We also see a selection of appropriate surrogate to establish AI. In order to have appropriate surrogate, the structure consideration should include whether nitrosamine structure alert is in the same chemical environment, the degree of substitution, the steric hinderings of the structure, electronic influences, potential metabolite activation, and the stability or reactivity of the resulting metabolite and overall metabolite molecular weight. In addition, the surrogate should have robust carcinogenicity data. We also see inappropriate application of molecular weight to justify the AI limit just through simple linear extrapolation. This is not appropriate because there's no sufficient data to indicate this is a linear, the molecular weight can linearly affect AI limit. The less than lifetime approach, LTL, has been applied to mutagenic impurities for those non-nitrosamine impurities in SHM7. Currently, this is under active research and evaluation to determine whether similar approach can be applied for nitrosamine impurities. And because it's still under current investigation, FDA do not accept this approach at this moment. Another similar issue is about derivation of AI limit from in vivo mutagenicity studies. This is also under current area for investigation, and therefore FDA currently do not accept this approach. And both topic actually will be included in SHM7 nitrosamine addendum development. So once FDA participate this effort, so once it's finalized, we will have update for that. And also I want to point out, SHM7 does not support the using in vivo mutagenicity study for mutagenic impurities at this moment. Also, more importantly, a positive mutagenicity result warrants a CPCA-based AI limit. We also see some applicants submitted repeated dose general toxicity to support AI limit. This is not only not appropriate, but also is not necessary, because such a study is not designed to evaluate the mutagenic or carcinogenic potential of nitrosamines. Instead, if you want to justify an AI limit above 1,500 nanogram per day, we recommend to conduct transgenic rodent assay to evaluate mutagenicity of the nitrosamines in vivo. So we also see some applicants just automatically apply SHQ3, Q3B, QT to control nitrosamines with negative in vivo mutagenicity study. As I just mentioned in the previous slides, FDA do not accept this approach automatically. We consider outcomes of compound-specific assessment and the patch testing results to recommend allowable limit. Some other inadequate justification also include setting AI limit recommended by other regulators without submitting the supporting data, or give FDA authorization for right of reference of those data. And also, it's not acceptable to support proposed control limit for impurity based on the level in the RLD. And also, as Dr. Vera explained, it's not acceptable to use interim AI limit as a final product control limit. So now let me present some case studies. The first study is using the surrogate compound. So the subject nitrosamine is category 3 compound with recommended AI of 400 nanogram per day, with molecular weight above 300 gram per mole. The applicant proposed AI limit of 1,000 nanogram per day. This is based on the proposed surrogate compound X. And FDA have a recommended AI of 1,000 nanogram per day for this surrogate compound. The justification included structure analysis indicate the nitrosyl structure alert in the surrogate was in the same chemical environment as the subject and the SRI. Although the surrogate has a lower molecular weight, the structure analysis also show both compounds have same degree of substitution, similar stereolytic hinderings and electronic influences, and have a similar potential for metabolic activation and similar reactivity of the resulting metabolite. So a review at the end accept proposed AI limit, and because we agree the surrogate S was an acceptable surrogate. This was based on the consult review from FDA's Computational Toxicology Consultation Service. Actually, their consult review identified 3 potential surrogates with sufficient structure similarity with the subject and the SRI. However, among 3 candidates, only surrogate S has robust carcinogenicity data and was selected as the valid surrogate. So the key point from this case study is a successful surrogate-based AI proposal needs thorough structure analysis and is supported by robust carcinogenicity data. So the second case study is about using compound-specific data. The subject nitrosamine is under category 2 with recommended AI limit of 100 nanogram per day, with molecular weight also above 300. The proposed AI limit was 1,500 nanogram per day. This is largely based on the submitted a positive 28-day in vivo transgenic rodent mutation assay using acceptable rodent models. In the analysis, the applicants derived a lower confidence limit of a benchmark dose that's from dose response of increased X gene in liver for its mutation frequency from controls. The benchmark dose, it is derived basically using a statistic modeling to get a point of departure from the dose response. Basically, below this dose, you will see negative effect for the examined endpoint. When you incorporate using the lower confidence, it's just incorporate the uncertainties in these studies. So it is a conservative approach. However, this approach applied to the in vivo mutagenesis data and just AI limit is currently under investigation. And also, more importantly, the submitted study indicate nitrosamine is mutagenic and is showing dose-dependent increase in mutation frequencies in relevant tissues at the top doses. Therefore, our review did not accept proposed AI limit. Instead, because the impurity is mutagenic in vivo, we recommend to control the impurity at a CPCA-based limit and also apply mitigation approach in order to achieve this limit. The key point from this case study is the variation of AI limit for nitrosamines from in vivo mutagenesis study is a current area of investigation. And therefore, FDA currently do not accept this approach. And this topic probably will be also incorporated into ICH-M7 nitrosamine addendum. So in summary, FDA nitrosamine guidance provide a framework to conduct risk assessment for nitrosamine impurities to set safe limit. To establish AI limit without compound-specific data, we recommend using CPCA as the starting point, as described in FDA real guidance. We also recommend applicant to monitor FDA guidance webpage for updates on control limits, as well as the safety testing approaches and other important informations. And also, I hope my presentation demonstrates that PharmTox in OGD works closely with key internal and external stakeholders to support end assessment, the guidance development, and the discussions to achieve international harmonization and nitrosamine safety risk assessment approaches, and as well advanced nitrosamine science. If there's no air limit for the nitrosamines of your interest, or you have a question to approach supported alternative limits, we recommend applicant to submit control correspondence to OGD to get most current thinking and recommendations from FDA. Here I provide some references that can be useful in nitrosamine safety assessment. And here I conclude my presentation. I also want to take this a moment to thank your attention for my presentation, and also thank you for the committee member for this workshop for their hard work. I also like to take this opportunity to thank my colleagues in the OGD's PharmTox group for their hard work and also help me with this presentation. Thank you.